Welcome to the deep dive. Today we're plunging into, well, the microscopic marvel that is your own body. Because, let's face it, life feels like a constant balancing act, doesn't it?
It really does.
But it's not just, on a grand scale, like walking that tightrope. It's happening deep within every single one of your cells, right now. As you listen, how does your body possibly orchestrate this intricate dance of building, breaking down, powering itself, all while keeping everything perfectly tuned? It's pretty mind-boggling.
It absolutely is. And that's exactly what we're tackling in this deep dive. Our mission really is to pull back the curtain on the fundamental energy reactions that drive all life, and to really understand how your body maintains its crucial internal stability, a concept we call homeostasis.
Right, homeostasis. And that's exactly our aim today, to be your guides, navigating you through what can often feel like, well, a complex biochemical landscape. We're going to try and break these topics down into digestible nuggets. Think like a first-year medical student. We use plenty of helpful anchors, maybe some acronyms, and a few questions to get your neurons firing.
Yeah, because understanding this foundational stuff isn't just academic, is it? It's the bedrock. It's what you need for diagnosing and treating disease later on.
Exactly.
Precisely. It's what everything else in medicine is kind of built upon. And speaking of the energy that drives everything, let's start with a thinking question for you, our listener. Something to mull over.
Okay, let's hear it.
Imagine you're about to run a sprint. Okay. Your muscles are suddenly demanding a huge, immediate burst of energy.
Right. Instant power needed.
What's the very first energy reserve your body taps into? And importantly, how long does it actually last?
Okay. First reserve. How long it lasts. Got it. Keep that in mind, folks, and we'll circle back to the answer later on. All right, let's dive straight into how your body manages this incredible balancing act. Starting with what we called its internal thermostat, homeostasis.
Homeostasis.
This isn't just some fancy word. It's the body's truly remarkable ability to maintain a relatively stable internal environment, no matter what's happening outside.
And what's fascinating here, I think, is that it's not static. It's not an unchanging state like a perfectly still statue.
Right.
Instead, we describe it as a dynamic equilibrium. Think of it like a tightrope walker. Constantly making tiny, almost imperceptible adjustments, maybe swaying slightly, but always returning to balance. It's not just standing motionless. It's an active process.
An active process. I like that. And what's truly profound here, maybe even a bit sobering, is that disease isn't always just an external attack. It's often an internal failure, isn't it?
Yeah.
A failure of these finely tuned homeostatic systems.
That's exactly right. The source material is very clear on this. Failure to maintain homeostasis leads to disease. It's that critical. And it operates at every single level, from the microscopic right up to the whole organism.
Every level? Like what?
Well, think cellular level. For example, the incredibly tight regulation of intracellular calcium ion concentration. Chi2 plus I.
Okay, why calcium?
Because it's absolutely critical for all sorts of cell signaling. Think muscle contraction, hormone secretion, neurotransmitter release. Tiny changes have big effects.
Gotcha. And then bigger scale.
At the tissue level, you've got this delicate balance between cell proliferation, making new cells, and programmed cell death, which we call apoptosis.
Apoptosis, right?
That balance is essential to keep tissues healthy and, prevent uncontrolled growth like cancer.
And then moving up again.
Organs, like your kidneys.
Yeah.
Constantly working, constantly regulating water and ion concentrations in your blood.
Yeah.
Filtering everything.
Yeah, the unsung heroes.
Totally. And then at the organism level, something we're all familiar with, your body maintaining that core temperature.
Yeah.
Around 37 degrees Celsius.
Yeah,
right?
roughly.
Regardless of whether you're out in a heat wave or stuck in a snowstorm, that's homeostasis in action.
It's incredible. All those systems humming along simultaneously.
It really is. And a classic example, one that medical students hit early on, is plasma glucose homeostasis. Blood sugar.
Yes, blood sugar. Crucial.
Your body maintains blood glucose in a pretty tight range, typically around 5 millimoles per liter. This is vital because glucose is the primary fuel for so many cells, but especially the brain.
The brain is picky about its fuel, isn't it?
Very sensitive to glucose levels, yeah.
And this is where the pancreatic duo comes in, right? Playing this vital role in a negative feedback loop.
Exactly. The stars of the show. When you're in a fed state you've just eaten, plasma glucose goes up, your pancreas releases insulin.
Okay, insulin's job.
Insulin basically tells your tissues, like muscle and fat, hey, take up more glucose. And it tells your liver to store excess glucose as glycogen. So it lowers blood glucose.
Brings it back down.
Brings it back down. Conversely, in a fasted state, maybe you haven't eaten for a while, plasma glucose declines, the pancreas releases the other hormone, glucagon.
The counterplayer.
Right. Glucagon signals the liver to do the opposite. Break down its stored glycogen and release glucose back into the blood, raising those levels.
It's such an elegant system, a real marvel of evolutionary design, optimized for, well, environments where food wasn't always guaranteed.
Precisely. But consider the modern irony, right? This finely tuned mechanism, designed for survival and scarcity, now often struggles to cope with an abundance of energy.
Leading to things like type 2 diabetes.
Exactly. The system gets overwhelmed.
It really paints a picture of incredible biological engineering. And speaking of levels and seeing what's going on, it's worth noting how doctors actually get clues about your internal state, isn't it? Because it's difficult, expensive, often painful to just go in and biopsy tissues directly.
Very invasive.
Yeah. So that's why measuring metabolites in your blood, things like glucose, amino acids, fats, cholesterol, urea, provides these vital diagnostic clues.
It's like a window.
A window into potential homeostatic imbalances. And here's a crucial tip, maybe the first big one for all you future clinicians listening. Always, always check your units.
Oh, absolutely. Crucial. A number without units is meaningless in this context. Millimoles per liter, milligrams per deciliter. It mattered.
I know, I had that drilled into me. It's fundamental.
It is. Blood is readily available. These tests are relatively inexpensive, making them indispensable tools. Deviations from the normal range is high or low. They point towards underlying issues.
Okay, so understanding this internal stability, this homeostasis, is one thing. But how does the body actually do all this work? That brings us right to the fuel, doesn't it? The world of energy and metabolism.
Yep. Every living cell needs a continuous supply of energy for work.
And when we say work, we mean lots of different things. It's not just lifting weights.
Not at all. To help remember the types of work, there's a handy acronym, BTMEO.
BTMEO. Okay, break it down for us.
Right. B is for biosynthetic work. That's building stuff. Creating new proteins after a workout, repairing cell membranes, making DNA, construction.
Makes sense.
T is for transport work. Think about your nerves firing constantly, pumping ions, sodium, potassium across membranes to send signals, moving things actively.
Oh.
M is mechanical work. Every twitch of a muscle, every beat of your heart, physical movement. E is electrical work. The very basis of thought in your brain involves electrical potentials across nerve cell membranes.
And O.
And O is osmotic work. Super important for things like kidney function, concentrating urine, moving water against gradients, maintaining fluid balance.
Wow, okay. BTMEO, biosynthetic, transport, mechanical, electrical, osmotic, all needing energy.
Constantly.
And where does this energy actually come from? You mentioned glucose earlier, but fundamentally.
Fundamentally, it's stored in chemical bonds. Energy is released when bonds are broken, or rather, when less stable bonds are replaced by more stable ones. And reactions that release more energy than they consume are called exergonic reactions. Think of breaking a stretched spring snap. Energy comes out spontaneously.
Spontaneous release, got it. And the opposite.
If a reaction uses more energy than it releases, it's an endergonic reaction, like compressing that spring. It needs an input of energy to happen.
Needs energy put in. Okay.
This brings us to a concept called Gibbs free energy, usually written as Ag delta G. Think of this as the usable energy from a reaction, the energy available to do work.
Usable energy.
Exactly. And the sign tells you what's happening. A negative G means energy is released. The reaction is exergonic, spontaneous, like ATP breaking down to ADP and phosphate that releases energy, so it has a negative G.
So if I'm getting this right, the cell cleverly takes energy from those spontaneous negative G reactions.
Yeah.
And uses it to power the ones that need energy, the positive G ones. It's like linking them together.
That's precisely it. That's the essence of coupling. It's how the cell gets things done.
Incredible efficiency.
It is. And this whole vast integrated network of chemical reactions in the cell, organized into pathways with starting points, intermediates, we call those metabolites, and endpoints, that's what we call cell metabolism.
Metabolism, the sum of everything.
Pretty much all the chemistry of life.
The metabolism has these two main teams working in tandem. You called them a dynamic duo earlier. Catabolism and anabolism.
That's right. Think of catabolism as the demolition crew.
Okay, demolition. Breaking things down.
Exactly. Catabolic reactions break down larger, complex molecules like glucose, fats, proteins into smaller, simpler ones. They're typically oxidative.
Meaning they involve losing electrons?
Losing electrons or hydrogens, yes. And they are exergonic. They release free energy. And importantly, they produce what we call reducing power. We'll come back to that.
Okay, so catabolism. Break down, release energy, reducing power. What about anabolism?
Anabolism is the construction team.
Building things up.
Precisely. Anabolic reactions synthesize larger, more complex molecules from smaller precursors. Think building proteins from amino acids or fats from fatty acids. They are typically reductive.
Getting electrons or hydrogens.
Yes, and they are endergonic. They require an input of energy, often using the energy released by catabolism, and they consume that reducing power that catabolism generated.
Ah, so they're completely linked. One provides the materials and energy for the other.
Interconnected and totally reliant on each other. You can't build without materials and energy, and you get those by breaking things down.
Okay, that makes sense. So given that your cells are constantly running both this demolition crew, catabolism, and this construction team, anabolism, here's another thinking question for you, listener.
Ooh, another one. Good.
What's the master architect? What tells the cell whether it should be breaking down fuel for energy right now or using resources to build new components? What directs the traffic?
The master architect. Good question.
Hold that thought. We'll definitely come back to that one.
Okay, let's delve now into the cells. Let's call it currency and couriers, starting with the big one, ATP.
ATP, adenosine triphosphate. You hear about it all the time.
You do. It's universally recognized as the cell's rechargeable battery, or maybe better, quick cache.
The cache, one out of battery.
Well, battery implies storage, and it's essential to understand ATP isn't really a long-term energy store. It's an energy carrier. It transports usable packets of energy right to where they're needed right now.
Ah, okay. So not the cell's big savings vault, but more like the readily available cash in its wallet, constantly being spent and replenished.
Exactly. That's a great analogy. And it does this through the continuous ATP-ADP cycle.
Right, ATP breaks down.
When ATP is hydrolyzed, meaning water is used to break off one phosphate group, it becomes ADP, adenosine diphosphate, and an inorganic phosphate ion we call pi. And crucially, this releases a significant amount of usable energy, about 31 kilojoules per mole under standard conditions.
31 kilojoule. Okay,
Yeah.
and that released energy powers.
It powers those endergonic reactions we talked about. Muscle contraction, active transport, building molecules, all that BTMEO work.
Got it. And it gets recharged.
And the beauty of the cycle is that ATP is then synthesized back from ADP and pi. This reaction requires that same amount of energy input, roughly 31 kilojoules per mole.
And where does that energy come from?
It comes from those exergonic catabolic reactions, the breakdown of glucose, the breakdown of fats. It effectively couples the energy-releasing demolition work to the energy-requiring construction and other cellular work. This coupling is fundamental.
So it's this constant cycle, break ATP, release energy, do work, use energy from fuel, remake ATP.
That's the core loop.
Right.
And while the majority of ATP, the vast majority in most cells, is produced through aerobic processes needing oxygen, specifically oxidative phosphorylation,
Right.
it can also be generated anaerobically without oxygen through a process called substrate-level phosphorylation. It's much less efficient, but absolutely crucial when oxygen is limited, like an intense exercise. Keeps things ticking over.
Okay. Now, is ATP the only player in town for this energy currency game?
Not quite the only one. There are other high-energy phosphate compounds in the cell, things like phosphenyl pyruvate, or PP, and 1,4,3-bisphosphoglycerate. These actually release even more energy upon hydrolysis than ATP.
More energy. Wow.
Yeah, they have even more negative G values. And they can directly transfer their phosphate group to ADP to make ATP. They're important intermediates in metabolic pathways like glycolysis.
Interesting. But ATP is still the main currency.
This is the universal currency, yeah. But this brings us neatly back to our sprinter.
Ah, the thinking question. Okay, I'm ready.
Remember the question. What's the very first energy reserve tapped into for that sudden burst?
Okay, the first one.
The answer is creatine phosphate.
Creatine phosphate. Okay, not ATP directly or glycogen?
Not first. Creatine phosphate acts like an emergency power bank, especially in muscles. It's capable of rapidly regenerating ATP from ADP, like boom, instantly.
So it's the immediate hero.
The immediate hero, but a short-lived one. It provides that huge power surge for only a few seconds, maybe 5 to 8 seconds of maximal effort. It's a very short-term store.
Okay, so creatine phosphate is essentially the body's turbo boost button. Perfect for that 100-meter dash, but definitely not getting you through a marathon.
Exactly. Unless you're incredibly fast, as you said.
So how does that work? How does it regenerate ATP so quickly?
Well, when skeletal muscle is at rest and ATP is abundant, an enzyme called creatine kinase uses ATP to phosphorylate creatine, forming creatine phosphate and ADP. So you build up a store of this high-energy creatine phosphate.
Stockpiling it during rest.
Exactly. Then, during that sudden burst of intense activity, when ATP levels start to drop because it's being used so fast, the reaction reverses. Creatine kinase takes the phosphate from creatine phosphate and slaps it back onto ADP, instantly regenerating ATP.
Wow. Simple, fast, reversible. Very clever.
Very. And here's the clinical relevance again, tying back to practice. Creatine and creatine phosphate eventually, non-enzymatically, break down into a waste product called creatinine.
Creatinine, I've heard of that blood test.
Exactly. Creatinine has no biological function, and your kidneys just filter it out and excrete it in urine. Now, the rate of creatinine production is pretty constant and directly related to your muscle mass. More muscle, more creatinine.
Okay, so it could be a rough marker for muscle mass.
It can. But more importantly, measuring creatinine levels in your blood and comparing them to levels in your urine is a crucial indicator of kidney function.
How so?
Because healthy kidneys are great at filtering creatinine out of the blood. If your kidneys aren't working well, creatinine builds up in the blood, so you see high blood creatinine. And less gets into the urine, so you might see low urinary creatinine excretion. It's a really key diagnostic marker for kidney problems.
Right, so that quick energy boost molecule leads to a waste product that tells us about kidney health. It's all connected.
It really is. Now let's shift gears slightly and talk about the electron dance of redox reactions.
Redox, oxidation and reduction.
Exactly. Biological oxidation, fundamentally, is about the removal of electrons, E, or often, in biology, entire hydrogen atoms, which is a proton, H plus an electron, from fuel molecules like glucose or fatty acids.
Okay, removing electrons or hydrogens, that's oxidation.
But these reactions don't happen in isolation. This is key.
Let me guess. They're coupled.
Always. All oxidation reactions are accompanied by reductive reactions. If something loses electrons, something else must gain them. They are always coupled and happen together.
Okay, and there's an acronym for this too, right? A classic.
The classic, oil rig.
Oil rig. Oxidation is loss.
Of electrons or hydrogen.
Reduction is gain.
Electrons or hydrogens. Simple, but effective. Helps keep it straight.
Definitely. So when fuel molecules are oxidized, where do those electrons or hydrogens go?
They don't just float off. They are transferred to specialized H-carrier molecules. Think of them as electron taxis or hydrogen shuttles.
Electron taxis, I like that. Who are they?
The main ones you'll encounter constantly are NAD plus 8, which gets reduced to NADH plus a proton, NADH plus H plus, FAD, which gets reduced to FADH2, and also NADP plus A, which gets reduced to NADPH.
NAD plus 8, FAD, NADP plus 8.
Okay. These molecules pick up the high-energy electrons, often as hydrogen atoms, from the oxidized fuels during catabolism.
Right, the demolition phase.
And they ferry this reducing power, these captured electrons, to other reactions, either to anabolic reactions that need them for building things, or ultimately, in aerobic respiration, they deliver them to oxygen via the electron transport chain, which drives massive ATP synthesis.
So they are crucial couriers, moving energy around in the form of electrons.
Yeah, absolutely vital.
You know, what often surprises me, and I think maybe listeners too, is where these vital carriers actually come from. They aren't just there, are they? There's a connection to what we eat.
Oh, absolutely. Another fantastic example of how interconnected it all is. These aren't magically synthesized from scratch easily. They're derived from B vitamins.
B vitamins, seriously.
Seriously. Niacin, that's vitamin B3, provides the core nicotinamide part of NAD plus and NADP plus. And riboflavin vitamin B2 provides the flavin part of FAD.
Wow. So your diet literally fuels these electron taxis. Deficiencies in B vitamins could actually impair your energy metabolism at this fundamental level.
Directly. It highlights why a balanced diet is so important for more than just calories. You need these micronutrients for the whole system to run properly.
Amazing. Okay, so we've got the currency, ATP, the couriers, NAD plus FAD, the energy release, catabolism, the building, anabolism. How does the fuel processing actually happen? You mentioned stages earlier.
Right. We can think of catabolism, the breakdown process, as generally proceeding in 4 main stages. It's a simplified overview, but helpful.
Okay, stage 1.
Stage 1. This happens largely through digestion. Dietary macronutrients, your carbs, fats, proteins, are broken down into their simpler building blocks, which become cellular fuel molecules. So carbs to glucose, fats to fatty acids and glycerol, proteins to amino acids.
Digestion and absorption. Got it. Stage 2.
Stage 2. These initial fuel molecules, glucose, fatty acids, some amino acids, are transformed within the cells into a smaller number of key metabolic intermediates. A really central one is acetyl-CoA. Some energy is released here, and some reducing power, NADH, is generated. Think glycolysis breaking down glucose to pyruvate, then pyruvate to acetyl-CoA.
Okay, converging on intermediates like acetyl-CoA. Stage 3.
Stage 3 is the big hub, the TCA cycle. Tricarboxylic acid cycle, you might also hear it called the Krebs cycle or the citric acid cycle, same thing.
The famous Krebs cycle.
Famous for a reason. Here, the acetyl group of acetyl-CoA is fully oxidized down to carbon dioxide, CO2. This releases a bit more direct energy, but crucially, it generates a lot more reducing power, lots of NADH and FADH too.
So TCA cycle is major source of those electron taxis getting loaded up.
Exactly. It harvests most of the electrons from the original fuel. And finally, stage 4.
Stage 4 is oxidative phosphorylation. This is where the payoff happens.
The big payoff.
All that reducing power, all the NADH and FADH2 generated in the earlier stages, delivers its high-energy electrons to the electron transport chain, which is embedded in the mitochondrial membrane. As electrons move down the chain, energy is released and used to pump protons, creating a gradient. Then, as protons flow back, they drive an enzyme called ATP synthase,
which acts like a molecular turbine cranking out massive amounts of ATP. This is where oxygen comes in as the final electron acceptor. It's highly efficient.
So break down intermediates, TCA cycle, loads up carriers, carriers drop off electrons, massive ATP synthesis using oxygen. That's the grand plan.
That's the core flow of aerobic energy extraction, yeah.
Which brings us back perfectly to our earlier thinking question.
Yes, the master architect.
What tells the cell whether to run the demolition crew, catabolism, or the construction team, anabolism? What's directing this metabolic traffic?
The answer lies in the balance of high and low energy signals within the cell. They act like metabolic traffic lights.
Energy signals as traffic lights. Okay, explain. What are the signals?
Think about what indicates the cell has plenty of energy. High levels of ATP itself, obviously, but also high levels of the reduced electron carriers. Lots of NADH, lots of NADPH, lots of FADH2. These are all high energy signals.
Just lots of cash, ATP, and lots of loaded taxis, NADH, etc.
Exactly. These signals act like a green light for anablism. They basically say, we've got plenty of energy and building blocks, time to build. So they activate anabolic pathways and often inhibit catabolic ones.
Makes sense. Use the surplus to build and grow.
Uh-huh.
So what are the low energy signals?
Conversely, what signals an energy deficit? High levels of ADP and AMP, adenosine monophosphate, which forms when ATP is heavily used, and high levels of the oxidized electron carriers, NAD plus O, NADP plus AAD, meaning the taxis are empty and waiting for fuel.
Lots of IOUs, ADP, and lots of empty taxis, NAD plus AH, etc.
Perfect analogy. These are the low energy signals. They act like a red light for building things and a green light for demolition. They activate catabolic pathways, break down fuel, we need more ATP, and inhibit anabolic pathways.
So it's this constant sensing of the cell's energy state, tuning the balance between breaking down and building up.
It's an incredibly elegant regulatory system. Ensures the cell always tries to meet its energy demands without wasting resources. It's almost humbling to realize this complex network is operating seamlessly inside us every second.
It really is. Okay, finally, let's talk about the body's strategic energy reserves. We mentioned the sprinter needing that instant creatine phosphate boost. But the body has plans for longer journeys too, right? Different fuel tanks.
Absolutely. There's a clear hierarchy of energy stores designed for different needs and durations.
So recap the super short term.
Very short term, lasting just seconds.
Yeah.
Creatine phosphate, right there in the muscle, instant ATP regeneration for that sprint or heavy lift.
Okay. What's next? What gets you through, say, a workout or a few hours between meals?
For immediate use, lasting minutes to maybe a few hours, depending on how hard you're working, your body relies on its carbohydrate stores. That's glycogen.
Glycogen, stored glucose.
Stored glucose, yeah. You have glycogen in your muscles, which is primarily used by the muscle itself for its own needs. And you have liver glycogen. The liver is more generous. It breaks down its glycogen to release glucose into the blood, maintaining blood glucose levels for the whole body, especially the brain.
So muscle glycogen is selfish, liver glycogen is generous.
Kind of, yeah. Think of glycogen as your daily drive fuel tank. Readily accessible, good for moderate duration.
Okay, but what if you need to go longer? Like days without food, or running an ultramarathon?
Then you tap into the long-term reserves. Your fat stores, primarily triglycerides stored in adipose tissue, fat cells.
The big tank?
The really big tank. Fats are incredibly energy dense. Gram for gram, they store more than twice the energy of carbohydrates or proteins. These reserves can last for days, even weeks, depending on the person, maybe up to 40 days or so. This is your long-haul flight fuel tank.
Incredible storage capacity. Is there anything else?
Well, there is one more, but it's really a last resort, used only under conditions of prolonged starvation or extreme metabolic stress.
What's that?
Muscle protein. Your body can break down functional proteins in your muscles into amino acids, convert some of them into glucose, or use them for energy. But this comes at a huge cost, loss of muscle function, strength, and overall tissue integrity. It's like siphoning fuel by dismantling critical engine parts. You really don't want to go there unless absolutely necessary.
Right. A desperate measure.
Yeah.
So a clear hierarchy. Instant boost, creatine phosphate, daily fuel, glycogen, long-haul tank, fat, and emergency breakdown protein.
That summarizes it nicely. So to kind of wrap up this deep dive, we've really journeyed through the core concepts, haven't we? We started with homeostasis, that vital dynamic equilibrium, the constant balancing act, keeping your internal environment stable.
Right. Not static, but dynamic.
Then we explored metabolism, this incredibly intricate, interconnected network of catabolism, the demolition crew, breaking things down to release energy and reducing power.
And enablism, the construction team, using that energy and power to build the complex molecules of life.
Exactly. We highlighted ATP as the universal rechargeable energy currency, the quick cash, not the long-term savings.
Constantly cycling through that ATP-ADP loop to power pretty much all cellular work, the BTMEO.
And we saw how the cell smartly regulates all this traffic, using those high and low energy signals as traffic lights, directing whether to build or break down.
Alongside having this amazing hierarchy of strategic fuel stores, ready for anything from a split-second sprint with creatine phosphate, to moderate activity with glycogen, to surviving for weeks on fat reserves.
And understanding these fundamental concepts, it really isn't just academic, especially for aspiring medical professionals. It's crucial.
How so? Practically.
Well, think about diagnosing metabolic diseases like diabetes. That's fundamentally a problem with plasma glucose homeostasis, or understanding a patient's nutritional and energy needs during recovery from major surgery or illness, or advising athletes on fuel for performance. These principles are woven through all of clinical practice.
They really form the bedrock, as you said earlier. It's this intricate, incredibly efficient, and, well, ancient system operating inside.
So ancient, fine-tuned over billions of years.
And that leads me to a final, maybe slightly provocative thought for everyone listening.
Okay.
Considering the body's remarkable efficiency in maintaining this balance, managing energy, all evolved in environments very different from ours, what might be the ultimate cost of our modern lifestyle? With its constant abundance of readily available high-energy foods and often much lower levels of physical activity on these finely tuned ancient metabolic pathways.
That is a really compelling question. What is the long-term cost of that mismatch between our ancient biology and our modern world?
Something to ponder, definitely. Until our next deep dive.